Hi guys, Dr. Lando here with another video. This one's on membrane transport. So there are two big categories for membrane transport, and they are passive transport and active transport. With passive transport, you're looking at diffusion, down the concentration gradient. That means we're going from an area of high concentration to an area of low concentration. Okay, high to low. This does not require energy. No ATP. Okay. Active transport, we are going against the concentration gradient. So we are pumping things opposite the direction they would like to go. Okay, and this does require ATP or some other form of energy, and there are others, even though I always talk about ATP. It does require energy, okay, to move the substances in and out. So we will look at both of these, passive and active transport. Okay, for types of passive transport, the first type of passive transport is simple diffusion. So with simple diffusion, small substances like water, carbon dioxide, oxygen move down their concentration gradient and go across the plasma membrane easily. Okay, so water, just this yellow line here, these yellow lines are representing my phospholipid bilayer, and water is just squeezing in between those phospholipids, no problem. Carbon dioxide, we could put him in there. Oxygen, any of those, they will go down their concentration gradients. Wherever they're high, they will go to low, and they will meet an equilibrium at some point. The other thing that can exhibit simple diffusion are things that are lipid soluble. So vitamins like A, D, E, and K, lipid hormones, any of the lipid-like structures that are small enough can go right through the plasma membrane. The plasma membrane is mostly lipid, dissolves like. They have no problem squeezing through right into the cell. They don't have to ask permission. They just say, well, excuse me, and walk right by the phospholipids. Simple diffusion, going down the concentration gradient. Next up is facilitated diffusion. So with facilitated diffusion, you need something to facilitate the diffusion. So let's get a color here. Let's go with purple. So out here we've got this high concentration of purple dots. What would this be? Well, it could be ions, for example. These could be chlorine ions or sodium ions, potassium ions, whatever. There's a bunch of ions out here. Notice that there is, we'll just put two of them in here. There's only two on this side. So this is a channel protein. Okay, that's a channel protein, and this channel is a happy place for these ions to go through. They are attracted to this channel, and then they are also attracted to going down their concentration gradient, so they want to go through here. So they will travel right through here. Why do we need this? Well, this is for any of those solutes that are either they don't need energy, okay? They might be too big to get through the plasma membrane, or they're not enough hydrophobic to get through the plasma membrane. They're too hydrophilic, so they have to have their own special channel to get through. So that's facilitated diffusion through a channel protein. Then we have this one over here. Let's give this guy a different color. Green. Okay. This guy, this is big. This can't possibly fit through the plasma membrane. So it attaches, this is called a carrier protein. Okay. So the molecule, the ligand, attaches on one side of the carrier protein. The carrier protein changes shape and releases the product on the other side. Again, we're going to go down the concentration gradient. This does not require energy, but it does require this protein, so it's facilitated. Okay, and the last type of passive transport that we talked about in class was an aquaporin. Need blue for this. Okay. I'll use this. So with an aquaporin, you're looking at water. And water can go across the plasma membrane by simple diffusion, but if you want a lot of water to be able to exchange, to go back and forth across the membrane more quickly, you give it a special channel. So this is a water channel specifically designed for water to be able to go down its concentration gradient, and it's called an aquaporin. Okay, so those are the three types of passive diffusion: simple diffusion through the membrane, facilitated diffusion using a channel protein or carrier protein, and an aquaporin special channel designed just for water. Okay, for active transport, the first one we're going to talk about is a protein pump. And that means that the pump itself is made up of a protein, not that it's pumping proteins. Okay, in this case, our pump is going to be pumping sodium and potassium. So here is a cell. This is the outside of the cell. This is the inside of the cell. So we've got extracellular space out here, intracellular space here, and I'm going to make my membrane go all the way. Okay, and we have to pump this sodium. This is what we call, and the sodium and the potassium. This is what we call the sodium-potassium pump. Okay, and this is a major ATP draw for the body. It's constantly working, pushing these ions against their concentration gradient. Notice that sodium is high outside the cell, low inside the cell. Potassium is low outside the cell, high inside the cell. And this pump is going to pump out three sodium for every 2 potassium it brings in. Okay, 3 sodium out, 2 potassium in. Just think of it like a little pump, like a sump pump in your basement that's pumping water out. This one just happens to pump in both directions. It pumps sodium out and pumps potassium in. When it does that, I kind of lost my arrow here, it does require energy, and that is a key characteristic of active transport. So it's going to take ATP, break it down into ADP and phosphate, and that's going to give the energy that is needed to the pump to do the pumping job. So that's the sodium-potassium pump, an example of a protein pump found in a membrane. It's an example of active transport. Requires energy and goes against the concentration gradient. 3 sodium out for every 2 potassium in. Uses 1 ATP for every turn of the sodium-potassium pump. Okay, here is a second type of active transport. This is endocytosis. We use this for bulk movement. Of particles inside. So bringing them inside the cell. Large particles, bulk meaning large. Big things that can't possibly get through a membrane protein or through the plasma membrane. So here's a little bacteria. I've got a big cell here. We'll say this is an amoeba. This is his nucleus here. And you got a little bacteria there. This is an amoeba. It's bacteria. And so these little arms, they're called pseudopods, reach out around the critter, and eventually they come together and pinch close, and you end up with the bacteria enclosed in a vesicle, or if this thing is planning on eating this thing, this becomes a food vacuole. Okay? That's endocytosis, bringing things in. There is another type of endocytosis that we talked about. We would call this actually phagocytosis. Okay, phago means to eat. And this critter, this amoeba, would be called a phagocyte, a cell that eats. Okay, another type of active transport, actually a type of endocytosis, is called receptor mediated endocytosis. Okay, so receptor mediated endocytosis would be something that would happen that the cell is supposed to take this item in. So these could be antibodies on the surface of a white blood cell, and they are there to pick up foreign substances, antigen. All right, so this, you could say that these little things are chickenpox virus, and the little blue dots are chickenpox virus, and the red sticks are antibodies against it. So the chickenpox virus attaches to the antibodies, and then this pit, it's a coated pit, it's coated with antibodies, begins to push in, and eventually you end up with needs to be a little thicker there, with a vesicle that is pinched off, and all of the stuff that you were trying to pick up, the cell was trying to pick up, is on the inside of the vesicle. Okay? So the virus was out here, it attached to the pit, the coated pit, the receptors. That pit began to invaginate, kind of fill with the weight of the virus, and then it eventually pitched off into a vesicle. All right, so the last type of active transport that we will talk about is exocytosis, and I talked about exocytosis a lot in this chapter. Probably didn't use the name exocytosis enough, so make sure that you know this is the name of the process, where a membrane-bound vesicle, that it contains something. We'll say that this vesicle contains an enzyme, was made at the ribosomes, packaged went through the ER, packaged at the Golgi, and now it needs to be released. So it gets moved to the plasma membrane. It will fuse with the plasma membrane and release its contents out into the extracellular space. Okay? So that's exocytosis. Moving, and the purpose of this is for bulk movement out of the cell. Okay, those are the types of membrane transport that you need to know for your exam. Make sure that you go through your notes and fill in all the little details that we talked about in class. And as always, if you have any questions, please feel free to contact me.